Let's begin with a fundamental question. What does it truly mean to age healthily? For years, science has grappled with separating the natural process of getting older from the diseases that so often accompany it. But what if we could quantify healthy aging itself? A recent study in Nature Aging has attempted just that, using machine learning to look beyond disease and identify the physiological signatures of longevity.
This brings us to a central problem in geroscience, the field dedicated to the biology of aging. You see, is a healthy older individual simply someone who hasn't yet received a diagnosis for a specific illness? For decades, this has been a substantial challenge: how to untangle the innate biological process of aging from the pathologies that frequently emerge alongside it.
Researchers often refer to this as the aging paradox. You can think of it as two intertwined threads. One is the biology of aging, and the other is the progression of chronic disease. In most individuals, these threads are so tightly woven together that they become nearly impossible to separate. Yet, being able to distinguish them is critical if we want to understand and promote a long, healthy lifespan. The dominant theory attempting to explain this connection is the geroscience hypothesis. It posits that a set of core, universal biological aging processes act as a primary driver for the development of many different age-related conditions, from heart disease to type 2 diabetes and chronic kidney disease.
So why is decoupling these two processes so difficult in practice? Well, there are several key reasons. First, for the majority of the population, aging and the onset of chronic conditions occur at the same time, which makes it very hard to distinguish cause from effect. Compounding this, the data we typically work with is incomplete. Most electronic health records only capture a fraction, perhaps 20 years of a person's life, which is insufficient to map the full trajectory from youth into old age.
To address these limitations, the researchers in this study developed a novel solution: a machine learning model designed not just to predict specific diseases, but to forecast an individual's entire health trajectory over their lifetime. The primary output of this model is a single, quantitative new metric. This new metric is termed the longevity potential score. It's a multivariate score, a single number that synthesizes many clinical variables to quantify a person's underlying physiological state, in essence, their potential for a long and healthy life. And critically, this is not just another way of expressing a low risk for disease. It is engineered to measure a separate biological phenomenon. The method used to construct this model is quite innovative. Instead of starting from a young age and predicting forward through time, it starts near the end of life and works backward. First, it learns to predict 5-year survival in 80-year-olds based on their recent health data. That prediction then becomes the target for a model trained on 75-year-olds, and so on, stepping back in 5-year increments. Each of these is a short-term prediction, but the model then stitches them together into a single probabilistic chain, what is known as a Markov model. The final result is a composite model that can forecast a person's lifelong trajectory from any age using only a few years of clinical data. The predictive capability of this score becomes evident when we examine the data. Let's take, for example, a cohort of 65-year-old women. Those individuals with a longevity score in the top 5th percentile have a 79% probability of surviving past the age of 85. In contrast, those in the bottom 5th percentile have only a 13% probability. This demonstrates a substantial divergence in outcomes predicted from routine clinical measurements. This leads to one of the study's most significant conclusions: the existence of what the authors term the longevity gap. It reveals that having a high longevity score is not simply the inverse of having a low risk for disease. It represents a distinct and previously unquantified dimension of health. To be precise, the model quantifies two different concepts. On one hand, it calculates the lifelong risk for 5 major chronic diseases. On the other, it calculates this separate longevity potential score. This score indicates a robust physiological state that is present even among individuals who already have a very low calculated risk for those specific diseases. And this is the core of the finding. Even within a group of people that current medical standards would label as healthy, meaning no current diagnoses and low calculated risk for future disease, there is a large, previously hidden spectrum of health. The model reveals a measurable variation in their underlying potential for healthy aging that was, until now, essentially invisible. The robustness of these findings is supported by the scale and breadth of the data. The model was initially developed using a comprehensive health database from Israel, which included 4.57 million individuals. Its predictive accuracy was then confirmed on 2 completely separate international cohorts, the UK Biobank and the US NHANES dataset. This demonstrates consistent performance across different populations and healthcare systems. So, which clinical markers are actually driving this longevity score, particularly within the strongly healthy population? The model was able to identify several physiological signatures, and some of them are not what one might conventionally expect. This table illustrates some of the key biomarkers. A high longevity score in healthy individuals was associated with mildly low neutrophil counts and low alkaline phosphatase, which aligns with conventional wisdom about inflammation and organ health. However, what is more counterintuitive is that the optimal levels for liver enzymes like ALT and for creatinine were not the lowest possible values. Instead, they were medium levels. This suggests that for healthy aging, the objective is not to simply minimize these markers, which in some cases could indicate low muscle mass or poor metabolic function. Rather, the key appears to be maintaining them within a specific, balanced, and optimal range. This quote from the research paper effectively summarizes the novelty here. The authors state, "The model we derived leads to a somewhat unexpected hierarchy of health levels in individuals who are currently considered strongly healthy. It shows that within a group we consider uniformly healthy, there is a quantifiable and predictive hierarchy of well-being that was previously unappreciated." Now, a critical question arises. Is this potential for longevity purely a product of lifestyle and environment, or does it have a genetic basis? The final section of the study connects this new health metric to genetics, providing compelling evidence that it is, in fact, significantly heritable. The most direct piece of evidence for this heritability can be distilled down to a single impactful number: a 1-year increase in the lifespan of an individual's parent. Let's clarify what this means. The researchers took the strongly healthy population and subdivided it based on their longevity score. They then compared the parents of the highest scoring group, called C1, to the parents of another group, C3, which was still considered healthy but had a lower longevity score. The finding was consistent across 2 different countries. The parents of the C1 individuals lived, on average, at least 1 year longer. The genetic analysis provides further support. Of 26 known longevity-related genetic variants, 17 maintained a significant association with parental lifespan, even when the analysis was restricted only to this strongly healthy population. This suggests a genetic signal for the healthy aging process itself, which is distinct from genes we typically associate with specific diseases. It also underscores the complexity of genetics. For example, certain variants of the APOE gene are well known to be linked to a higher risk of Alzheimer's. Yet this study found that independent of that risk, other variants in that same genetic locus are associated with a longer parental lifespan, demonstrating how a single gene can have multiple and sometimes opposing effects on health. In conclusion, this research provides a new quantitative tool for deconstructing the aging process. By defining and measuring a dimension of health that extends beyond the mere absence of disease, it opens up new avenues for both research and potentially preventative medicine. It leaves us with a compelling question for the future. If our potential for healthy aging is encoded in our physiology long before disease becomes apparent, how might we leverage this knowledge to promote longevity for a wider population?